Hi guys, this video is about gene regulation. In the video I'll talk about what gene regulation is, and then I'll give some examples of how it happens. There'll be two sets of examples. One that happens before transcription, so before DNA is transcribed into mRNA, and some that happen after transcription, so after that mRNA has already been made. So gene regulation is your cell's ability to turn on or off certain genes depending on the need of the cell. So not all of your cells express all of the DNA that they have, because all of your cells have all the same DNA. Your bones and your muscles have the same exact DNA, but they're very different because different genes are expressed in them. So for example, in your neurons, in the nerve cells in your head, they're not making much keratin, which is a protein that makes your hair and your nails strong, but nerve cells don't have any need to transcribe the DNA, the gene for keratin, and in order to make that protein, because they don't need that protein. We have about 200 types of cells in our body, and like I said, they all have the same DNA. The reason that the cells can be different is because of differential gene expression. So different genes are expressed in different cells. Most regulation happens at or before transcription, so either prevents transcription or speeds up transcription or in some way influences that step in the process. So there's two major classes of how genes are regulated at the transcription stage. One major class is the regulation of chromatin. So remember, chromatin is what we call DNA when it's all wrapped up in the proteins that keep it organized and condensed in a nucleus. So chromatin is made up of DNA and proteins. DNA is blue here. The proteins, specifically proteins called histones, are pink in this picture. So there are two types of chromatin regulation. We can either regulate the DNA or regulate the histones. Histone modifications are when we're making changes to these proteins to change where the DNA is, so it can be transcribed or not transcribed. So what do I mean by that? DNA is wrapped around these histones. The parts of DNA that are exposed, that are between histones, can be transcribed. Transcription factors and RNA polymerase can get in there and do what they need to do to copy that gene. But DNA that's wrapped around the histone is not as accessible, and so is not transcribed. So one of the ways that your cells regulate what genes are expressed and are not expressed is by where the DNA with that gene is relative to histones. But the location of histones can be changed through something called histone acetylation. So what that means is an acetyl group is added, I forgot the L, acetyl, can be added to histones, and when those are added, it ends up changing how the histone is associated with the DNA. So it could uncoil the DNA, the histone could move, but it ends up exposing DNA that was not exposed before so that it can now be transcribed. Another form of chromatin regulation is DNA methylation. So we just talked about changing the proteins in the chromatin. Now we're talking about the DNA. DNA can have methyl groups added to it, so CH3 additions of those extra atoms of CH3 groups. If the DNA is methylated, if there's a lot of these methyl groups attached to it, transcription doesn't happen. If there are not methyl groups attached, transcription does happen. So this is another way that cells regulate what genes are transcribed and what genes are not transcribed. Methylation, no transcription. No methylation, transcription. This can be changed. Methyl groups can be added or removed based on what genes need to be expressed. Both DNA methylation and histone modification are two regulation of chromatin mechanisms are part of something called epigenetics. So this is the study of modifications to chromatin and how they influence what genes are expressed. What's really cool about epigenetics, and there's a lot of research happening about this now, is that there's evidence to suggest that your epigenetics, your DNA modifications can be passed on from parent to offspring. So if I, in my cells, had methyl groups on one sequence of DNA of a specific gene, that could be passed on to my offspring. So scientists are still studying how that is and why that is and how it's passed on and all that stuff, but it's pretty cool. So that was all regulation of chromatin. Now we're going to talk about regulation of transcription itself. When we learned about transcription, we talked about promoter regions on the DNA, and we talked about how transition initiation complexes form and attach to the promoter, and that's how transcription is initiated. So remember, those initiation complexes contained transcription factors and RNA polymerase. We're going to get a little bit more detailed now. There are more things happening than just the initiation complex forming onto the promoter. There are other regions of the DNA that are important in whether or not that gene is going to be transcribed and eventually expressed. There are regions called enhancers, which are actually way, way, way upstream of the gene itself. This line is supposed to be like in a graph. If you have like on an axis that you need to show there's a big jump in numbers, you put kind of like a squiggly line. That's what this line's supposed to be. So enhancers are sequences of DNA that are way upstream that can influence whether or not a gene is expressed. And control elements are other sections of DNA that are closer to the promoter that also can influence whether or not a gene is expressed. The way this happens is transcription factors, just like we talked about with the transcription initiation complex. They combine and attach to other proteins and RNA polymerase and can either encourage transcription or prevent transcription. If a transcription factor binds to DNA and it activates transcription, transcription happens. We call it an activator. If when a transcription factor binds to DNA, transcription is not happening, that's called a repressor. It's repressing that gene. There are many different transcription factors, and there are many different ways that they can bind together to influence the expression of different genes. So those were all gene regulation mechanisms that happen before transcription, and now I'm going to talk about some post-transcriptional modifications, so modifications that happen after that mRNA is transcribed from the DNA. One is mRNA processing. We've already talked a lot about this. The mRNA, the pre-mRNA, is given a 5' methyl cap, 3' poly A tail, and the introns and exons the introns are spliced out, exons are kept, and alternative splicing can happen. So exons can be combined in different orders. So that's one way that transcription can be modified. Gene expression can be modified. So a couple other ways that gene expression is modified after transcription are mRNA degradation. So this means that mRNA is broken down in the cytoplasm by enzymes. This can happen at varying rates. So that's another way that gene expression can be influenced, because if you break down all the mRNA for that protein, for that gene, the protein can no longer be made. Okay. And the next one, initiation of translation. So when we learned about translation, we learned about this step of initiation that starts the process. Initiation of translation can be interrupted or encouraged, I guess, or enhanced in two basic ways. It can either be targeted to one specific mRNA sequence, and so one specific protein, or it can be global, meaning all across the cell, all protein synthesis, all translation is scaled back or cut down. That would be by controlling like the amount of ribosomes that are present, or the amount of tRNA that are present, or other ways of messing with the tools that are needed for translation. There's also protein processing and degradation. So proteins, after they're made from ribosomes, by ribosomes, they undergo chemical modifications, kind of like the DNA and the chromatin that we saw. Acetyl groups, methyl groups, other groups can be added to the protein, and the protein is folded in the way that it's supposed to be into its 3D structure. Also, there are proteasomes, so enzymes that break down proteins, and there can be more or less of them depending on if the cell needs that protein to stick around for a long time, or doesn't want that cell or doesn't need that protein to stick around for a long time. But that would be the degradation aspect. So just to summarize, gene regulation, what is it? It's whether or not a gene is expressed in a cell, because not all cells express all the genes. And we talked about before transcription regulation, and we talked about after transcription regulation. Thanks, guys.